So first we're going to draw the pre and post synaptic terminals. And these synaptic terminals are where nerve cells are communicating, or cells in general are communicating. And then from here, we will have a few vesicles coming from the presynaptic terminal. And in those, they will be containing a bunch of little blue dots that we will touch on a little bit later. Also inside of this, we will have organelles that help with cell communication. We will have receptors for ligands. And pathways that help release these ligands. And the same will be true for the postsynaptic terminal. So these blue things, they are adrenal receptors, and the pathway is this right here is a open vesicle that also contains one of these. We have one of these proteins that is MAO. We have a mitochondria right here. And this protein is COMT. And MAO and COMT will be used to help transport these signals and amplify them from our ligands. So these will come in from here. That would be an ion-gated receptor, and this is also going to go in here, but this is going to stay in here, and it is going to give us an output that'll be used for the cell. And from this cell, from this information here, it's going to go through this over here. It's also going to go up through this protein so that we can get some of these little micro monoamines down into this area where we can form them in the vesicles and get them out into the intermembrane space. And when out in the intermembrane space, these are going to go and attach onto these, where they will bond with G protein receptors or another type of receptor. And after that, they will turn into kinases or turn into cyclic AMP which will activate protein kinases. And this is all for a normal cell. So if we draw the same picture again, and we get rid of this right here, and we draw another vesicle that is emerging. And say we add a few more of these. Oh, there you go. This is going to be, as we said before, the presynaptic gap. Or presynaptic terminal. And this is going to be the postsynaptic. Again, these are nerve cells. And this is for a cell that would be under the presence of cocaine. So just to get we have some vesicles within the cell that are going to contain different proteins or different types of ligands and communication devices that the cells can use. And then we will also have protein channels that can help all throughout the cell. And we're gonna make a new color. We're gonna say this pink stuff is cocaine. So these pink things are going to block up these channels so that when we release our molecules and our ligands, they cannot go back here into these receptors. So they cannot go in there. This cannot go in here. And then this cannot go in here. So these receptors are being blocked. So we have an excess amount of all these things within the inner membrane space, or synaptic cleft. This thing right here is called a synaptic cleft. Which is just the space in between axon terminals and dendrites. And then we have each individual receptor protein that we're going to use as these squiggly lines. And we have the center one is going to be a dopamine receptor. This is going to be an adrenoreceptor. And this will be a serotonin. And what we can see is with the presence of cocaine, there's an increased amount of serotonin, adrenaline, and dopamine within the inner membrane space, and they are all going to be going towards these detectors in other cells. And these are all going to, draw arrows over here, these are all going to result in different psychophysiological effects that are the common effects known to be from cocaine.